Cell biology and biotechnology. We already studied the main aspects of the biotechnology with the help of microorganism and technology. Scientists, professors produces the products which are useful for human being or mankind. Human health includes the various diseases, and these diseases can be treated with the help of vaccines. Various vaccines and antibiotics are also produced in the same way. Vaccines and antibiotics are produced in the laboratories with the help of some microorganism or with the help of some animals. Here, the father of antibiotic is Alexander Fleming. First antibiotic produced in the laboratory, that is the penicillin, from the fungus Penicillium. Who is the father of antibiotic? Alexander Fleming. Here, next. This is the culture media, this is petri dish, and this is the growth of the microorganism which are used for production of antibiotic. Vaccines and vaccination. Here polio and other some diseases are treated with the vaccines like tell me the example of vaccines. Polio vaccine, hepatitis vaccine and tetanus vaccine. These are the examples of vaccines. Vaccine is the antigen containing material given to acquire either permanent or temporary immunity against a specific pathogen or disease. What is meant by vaccine? Vaccine is the antigen. And what is meant by antigen? Antigen is the substance which can stimulate the body for the production of antibody. Here antigen is the foreign substance and antibody produced inside the body by the WBC. WBC, these are white blood cells, produces antibodies for the immunity. When immune system is strong, there is no antigen work. Here, antigen comes from outside. Antigen, natural bacteria, this is causing bacteria, this is causing viruses, this is causing fungi, this is causing any chemical molecule. These act as an antigen. Against that antigen in the body, antibodies are produced by the WBC in the blood. When there is an antigen coming in contact with the antibody, there is an antigen-antibody complex get formed and antigen is neutralized by the antibody or destroyed by the antibody, and yes, body is protected against the disease causing antigens. This is the method or mechanism of antigen-antibody reaction. Immune system, if immune system is strong, yes, no any disease can cause disease to our, no any pathogen cause disease to our body. Therefore, immune system should be strong. Vaccine is the antigen containing material given to acquire, either given to person, to acquire, given to person or human being to acquire, either acquire in Marathi प्राप्त करणे, either permanent कायमस्वरूपी or temporary म्हणजे तात्पुरती immunity against a specific pathogen or disease. Specific विशिष्ट रोगाच्या विरोधात प्रतिकार क्षमता तयार होण्यासाठी जर अँटीजेन म्हणून आपण इंजेक्ट केलं किंवा अँटीजेन म्हणून जर काही वॅक्सिन दिलं, काय प्राप्त करण्यासाठी? परमनंट किंवा तात्पुरती किंवा टेम्पररी इम्युनिटी प्राप्त करण्यासाठी बाहेरची वॅक्सिन किंवा ज्या वॅक्सिन मध्ये अँटीजेन आहे असे घटक दिले तर आपल्या शरीराला ट्रेनिंग मिळतं. Means what? When vaccine enters into our body, prepare for, yes, destroy the preparation or training of the WBC is due to previous, yes, identification of this antigen, and previous knowledge helps to remove or destroy real when real antigen enters into our body, what happens? That antibodies comes in action immediately and destroy that antigen. Therefore, after some time of vaccination, there is a, if any attack of the any disease causing virus or bacteria, disease does not occur, because this is action of vaccine is like a, this is the mechanism of vaccination. What happens if there is a previous vaccination is there, real antigen of that disease does not disturb your body, because already antibodies are ready to destroy the antigen, and therefore no disturb or no disease-causing microorganisms like bacteria and viruses does not cause any harm to our body. Next.
Containing material given to acquire either permanent or temporary immunity against a specific pathogen or disease.
Traditionally, vaccines were prepared with the help of pathogens. Have killed a microorganism or have killed a pathogen of that disease is nothing but vaccine. Completely or partially killed pathogens were used as a vaccine. Paralyzed or completely killed or completely disturbed genetic material only. The genetic material which is present in the pathogen should be deactivated. Therefore, there is no disease-causing ability to that microorganism. Partially killed or completely killed microorganism of that disease is called as vaccine. Okay?
With the help of pathogens, partially killed pathogens were used as vaccines.
As an alternative, scientists tried to artificially produce vaccine with the help of gene isolated from the pathogen and used it as vaccine. That means in laboratories, alternative. Scientists tried to artificial production of vaccine. Nowadays we use animals or microorganisms for the production of vaccine, but in the laboratory nowadays artificial production of vaccine with the help of gene isolated from the pathogen. That means any disease causing pathogen is taken and gene is isolated and that gene used for production of vaccine in the laboratory. That gene does not cause harm to, but only having capacity to stimulate the body for the production of antibody. Stimulate the body for production of antibody. It acts like a antigen, but it is not really antigen. Okay?
Still try to artificially produce vaccines with the help of gene isolated from the pathogen and use it as vaccines. Then,
safer vaccines are produced in the laboratory.
Now, proteins which act as antigens are injected in pure form instead of injecting the killed or semi-killed parts.
Just wait. Nowadays, proteins which act as antigen are injected in pure form instead of injecting killed or semi-killed pathogen. Instead of using microorganism of that disease, half killed, we can use only proteins in pure form which act as an antigen, which is produced inside the laboratories.
These proteins keep people away from the diseases by keeping the immune system active.
These proteins keep the people away from the diseases by keeping the immune system active. What happened? Immune system always kept active due to this antigen proteins. Okay.
The vaccines produced with the help of biotechnology are safest, more thermostable, and remain active for longer duration.
What are the advantages of artificially produced vaccines? Artificially produced vaccines are more thermostable. They can be kept for long period into the laboratory, into the market, pharmaceuticals, etc. They remain active for longer duration in the hospitals. म्हणजे ह्या लसी आपण खूप दिवस हॉस्पिटलमध्ये ठेवू शकतो, नाहीतर डिस्कार्ड करावा लागेल. Example vaccines of polio and hepatitis.
Vaccines of polio, hepatitis. Edible vaccines.
Edible vaccines. Yes, work on production of edible vaccines is in the progress and presently potatoes are being produced with the help of biotechnology. Nowadays, we already seen that biotechnologically transgenic potato helps in boosting our immune power.
These potatoes are called transgenic potatoes.
These are transgenic potatoes.
These potatoes will act against bacteria like Vibrio.
Against which bacteria they act? Vibrio coli and Escherichia coli.
Consumption of these raw potatoes generates immunity against cholera and disease caused
due to E. coli. Okay.
B. Treatment. Biotechnology is useful.
Biotechnology is useful for the production of hormones like insulin, somatotropin. blood clotting factor. Okay.
somatotropin, and blood clotting factors. C. Interferon. This is a group of small-sized protein molecules used in treatment of viral disease. These are
Interferons, that is the group of small sized protein molecule used in the treatment of viral diseases. These are produced in the blood. Here, interferon. These are viruses. When viruses enter into the cell, living cell, they replicate. Viral replication inducing stimuli. Okay. And after stimulation, they produce inhibition of viral replication. Here, IFM produced antiviral protein. When interferons are produced against viruses, they stop the replication of viruses inside the cell. Viruses are in small entity, very small entity, which are on the threshold of living and non-living things. When they enter into living cell, then and then only they replicate. Therefore, viruses, when viruses enters into the cell, due to stimulus of virus, interferons are produced for the protection inside the cell, and when interferons are, when injecting artificial interferon, then what happens? Replication stops.